Intricacies of Polycystin1 Processing and Biochemistry. In this video, we will discuss the structure of polycystin proteins and describe the molecular mechanism of polycystin-1 cleavage at a G-protein-coupled receptor proteolysis site, or GPS. Describe the biochemical complexity of polycystin-1 generated by the cleavage reaction, and discuss Western blot biochemical analysis of the full complement of polycystin-1 protein molecules. Autosomal dominant polycystic kidney disease, or ADPKD, is a multisystem genetic disorder characterized by kidney cyst formation that can begin during fetal development and continue throughout life, eventually leading to end-stage kidney disease, or ESKD, in approximately 50% of those affected. ADPKD is the fourth leading global cause of end-stage kidney disease. ADPKD occurs due to mutations of two main causative genes, PKD1 on chromosome 16, which encodes for polycystin-1 or PC1 protein, and PKD2 on chromosome 4, which encodes polycystin-2 or PC2 protein. PKD1 gene mutation is more common and accounts for about 78% of the families with ADPKD. PKD2 accounts for about 15%. In about 7% of the families, neither mutation can be found, noted as NMD. Polycystin-1 and 2 proteins localize to primary cilia of the renal tubular epithelial cell and regulate cell-cell interaction and function. Cysts are formed in ADPKD kidneys when the activities of the polycystin proteins fall below a threshold level. Now let's learn about the structure of the polycystin proteins. Polycystin-1 is a 4,302 amino acid, 11 transmembrane receptor-like glycoprotein with a large extracellular region. The G-protein couple receptor proteolytic site, GPS, is situated at the base of this region. This is the site where the polycystin-1 protein is cleaved, which results in the formation of two fragments, the N-terminal fragment, or NTF, and a C-terminal fragment, or CTF. The NTF contains numerous domains that are involved in protein-protein or protein-carbohydrate interactions. Polycystin-2 is a 110 kilodalton protein containing 6 transmembrane spanning domains with intracellular C and N termini. It's a member of the transient receptor potential TRP channel family. Polycystin-2 properties allow it to form a homotetrameric complex that likely functions as a non-selective cation channel. Additionally, polycystin-2 can interact with polycystin-1 to form a heteromeric complex with a proposed stoichiometry of 1 polycystin-1 molecule to 3 polycystin-2 molecules. This interaction is thought to occur via coiled domains within their C-termini and between their top domains. Whether this heteromeric complex also has channel activity remains an area of active investigation. Now that we understand the structure of polycystin proteins, let's discuss polycystin-1 cleavage. A fundamental property of polycystin-1 is cis-autoproteolytic cleavage, that is, cleavage via a self-catalyzed chemical rearrangement without requiring exogenous proteases. As mentioned before, this cleavage occurs at the GPS site, which is situated at the base of the extracellular region and approximately 20 amino acids upstream of the first transmembrane domain at the amino acids HLT as shown. The resultant fragments, polycystin-1 NTF and polycystin-1 CTF, have a complex interaction that will be discussed a bit later in this video. The GPS motif is a defining feature of adhesion G-protein couple receptors, or AGPCRs. GPS has recently been recognized as a part of the larger GPCR autoproteolysis-inducing domain, also known as gain domain, that is present in polycystin-1, shown here in dark blue. The cleavage process is necessary to promote ciliary trafficking and full function of polycystin-1. The functional significance of GPS cleavage is highlighted by the finding that PKD1 GPS mutant mice develop polycystic kidney disease. Approximately 30% of all known pathogenic missense and small deletion insertion mutations are located in the gain domain and are predicted to disrupt polycystin-1 cleavage. Now, brace yourself for some intense chemistry to understand the proposed mechanism of polycystin-1 protein cleavage. Here is a magnified image of the GPS motif. The cis autoproteolytic cleavage of PC1 occurs at a conserved HLT tripeptide within the GPS as shown here. The actual cleavage occurs between leucine, marked as L, and threonine, marked as T. Cleavage of polycystin-1 at the GPS site occurs constitutively and extensively and takes place in the endoplasmic reticulum shortly after protein synthesis. The threonine residue of the HLT tripeptide initiates a nucleophilic attack. This results in the formation of a tetrahedral intermediate. This then undergoes an NO-acyl shift, which converts the peptide or amide bond to a more reactive ester intermediate. Hydrolysis of the ester bond leads to polycystin-1 cleavage, generating NTF and CTF fragments. The details of this cleavage are complex and are discussed more in the reference below. It's important to emphasize that polycystin-1 proteolytic cleavage is incomplete, irreversible, and the extent of cleavage is variable. After cleavage, the two resulting fragments, polycystin-1 NTF and polycystin-1 CTF, remain tightly and non-covalently associated to form a heterodimeric molecule. In summary, GPS cleavage generates biochemical complexity of polycystin-1 molecules, which in turn complicates protein analysis. Here we show three major polycystin-1 forms that exist in tissues and cells. Form 1 is the uncleaved polycystin-1 that is present in a small amount and is restricted to the endoplasmic reticulum. Form 2 is the cleaved heterodimeric, non-covalently bound full-length polycystin-1 comprised of PC1 NTF and PC1 CTF. This is the predominant form in most tissues, which traffics to the primary cilium. PC1 NTF can be disassociated from the heterodimeric, non-covalently bound PC1 molecule and becomes an independent or detached form of polycystin-1. This is form 3, which may localize to the plasma membrane of renal epithelial cells. Of note, the remaining polycystin-1 CTF fragment is not detectable, and its fate is unknown. Analysis of the full complement of polycystin-1 products requires the use of a combination of specific antibodies directed to PC1 NTF or PC1 CTF. There are many antibodies that have been developed for detection of polycystins 1 and 2. Many of these are available through the PKD RRC website. Here is the link. Antibodies that are directed against one cleavage fragment of polycystin-1 cannot detect the other cleavage fragment by SDS polyacrylamide gel electrophoresis, also known as SDS PAGE. Therefore, to fully analyze the polycystin-1 products in the cell, immunoprecipitation is required. For example, in this slide, we have generated total lysate from cells. The total lysate is immunoprecipitated with an antibody directed at the C-terminus of polycystin-1 or anti-CTF. This antibody will be able to immunoprecipitate forms 1 and 2, whereas form 3, due to lack of the CTF fragment, will come through in the flow-through fraction. The IP products are analyzed on a denaturing gel, and the blot can be probed with either an anti-CTF antibody or an anti-NTF antibody. Of note, the denaturing gel separates form 2 into its individual protein fragments, NTF and CTF. Therefore, the anti-CTF antibody will detect form 1 as well as the separated CTF fragment of form 2. The blot can also be probed by an anti-NTF antibody, which will detect form 1 and the NTF fragment of form 2. After completion of polycystin-1 cleavage in the endoplasmic reticulum, the protein undergoes additional N-glycosylation as it traffics through the Golgi on the way to the cilium or plasma membrane. Glycosylation status can be probed by treating lysates with PNGase F or endo-H noted by a P and E, respectively. PNGase-F efficiently removes almost all N-linked oligosaccharides from glycoproteins. If a protein is glycosylated, it will appear as a smaller band on the gel once oligosaccharides are removed by PNGase-F. The activity of endo-H is more specific. This enzyme cleaves between 2N-acetylglucosamine residues in the core of an oligosaccharide. As membrane proteins traffic through the Golgi, they become resistant to the activity of endo-H due to additional modifications. Most of the cleaved full-length PC1 molecule is endo-H resistant, but there is a pool that remains endo-H sensitive. Here is an example of a Western blot showing N-glycosylation analysis. Corresponding on the right is a cartoon of the same. Cell lysates are immunoprecipitated with anti-CTF antibodies. The IP products are either untreated, indicated by U, or treated by PNGase-F, or endo-H, indicated by P or E, respectively. Because polycystin-1 is almost entirely cleaved, the most prominent band visible is the PC1 CTF. A very small amount of uncleaved polycystin-1 is visible at the top of the gel. All of the sugars on the PC1 CTF are removed by PNGase-F, resulting in a single, smaller band in the center. In contrast, treatment with endo-H results in two bands, the top one representing the endo-H resistant fraction noted by the letter R. This is better visualized on the less exposed blot on the left. The blot can also be probed by anti-NTF. Here in the untreated or U lane, we see the uncleaved polycystin-1 in red and two blue bands representing the glycosylated forms of NTF. Treatment with PNGase results in two bands, a top faint red band, which is the unglycosylated form of full-length polycystin-1, and the lower blue band represents the unglycosylated NTF. Treatment with endo-H reveals that a large fraction of the NTF is endo-H resistant. Increased exposure of the blot shows a small amount of endo-H sensitive NTF. In summary, autosomal dominant polycystic kidney disease, or ADPKD, occurs due to mutations in two genes, PKD1 and PKD2. The protein products of these two genes, polycystin-1 and polycystin-2, interact with one another to form a complex. Polycystin-1 is a large transmembrane receptor that undergoes autoproteolytic cleavage at a GPS site. This results in two fragments, the N-terminal fragment, or NTF, and a C-terminal fragment, or CTF. The NTF and the CTF remain non-covalently associated to form a heterodimeric molecule. Polycystin-1 cleavage at GPS is incomplete and variable and results in considerable complexity with multiple polycystin-1 forms. Analysis of the full complement of polycystin-1 forms requires a combination of immunoprecipitation and Western blot using antibodies directed to NTF and CTF. Polycystin-1 is highly glycosylated as it traffics through the Golgi on the way to the primary cilium or plasma membrane, resulting in additional complexity on Western blot. Glycosylation status can be probed with PNGase-F or endo-H. This brings us to the end of this video.